Hello, everyone. My name is Phatak Farahani, Marketing Manager at Advanced Cell Diagnostics, and your host for today. Welcome to ACD's Knowledge Resource webinar series. We will give you closer understanding of how Autoscope Technology is contributing to specific application areas. We begin our knowledge series today on the topic of pain medicine, presented by Jyoti, Phatak, Sheldon. Jyoti holds a master's degree in molecular and cell biology, along with a bachelor in dental surgery. She has previously worked in the field of genomics and precision diagnostics as a product specialist for qPCR-based liquid biopsy tests. Jyoti currently works at ACD as an associate scientist in the application team to execute projects and collaborations that demonstrate novel applications of RNAscope, BaseScope, and microRNAscope assays. We'll now begin. Thank you. Jyoti?
Thank you, Phatak, for the introduction. And thanks everybody for joining today's Knowledge Resource webinar. I hope you can all hear me okay. So in today's Knowledge Resource webinar, my focus is going to be on showing you how you can apply our technology in pain medicine research. There's been a lot of uptake of publications and research funding for study of pain medicine and pain therapeutics. And I really wanted to kind of highlight this niche group of research area to all our existing users or potentially new users. So let's first understand what Autoscope technology is. The Autoscope technology is an ideal spatial solution. So it's really allowing you to interrogate those complex and highly heterogeneous tissues, but on that spatial and the morphological level. The novelty of this assay is it gives you that highly specific and sensitive method of detecting RNA biomarkers in both cells as well as tissues, once again with that morphological context, but really showing you all of this data at that single cell level. Now, this technology consists of three parts, or the technology's novelty is based on three parts. The first is its unique target probe that ACD will design against your sequence of interest or your gene of choice. Then second is our signal amplification system. And that is the one that's really allowing you to generate that high signal to noise ratio. And lastly, you can get to visualize your data as the single RNA molecules or dots. Now, this assay will allow you to spatially map different kinds of RNAs like mRNAs, long non-coding RNAs, splice variants, specifically highly homologous sequences that are very challenging with traditional ISH techniques in both cells as well as in intact tissues. And you can look at all of this with either a fluorescent or a chromogenic label. Our assay can be performed on a wide variety of sample types. Now, depending on the subcategory of the assay you choose, either the chromogenic assay or the fluorescent assay, the singleplex or multiplex assays, you can test this assay on either FFPE tissues, fresh frozen or fixed frozen tissues, PBMCs, or even cultured cells. So we talk about this assay having that high sensitivity and specificity. And how are we really achieving that? So the two key features of this technology, as I mentioned before, is its probe design and its signal amplification. So you see here is that oligonucleotide target specific probes that are designed by ACD, you see are depicted as these Z's. And it is to emphasize the fact that these two regions are linked by a spacer. Now this bottom of this Z is complementary and hybridizes to your target transcript or your gene. While this top of the Z, this creates a base for that building of the amplification structure. So now when these two Z's hybridize in tandem on the target sequence, it creates this binding site upon which this preamplifier can be built, and then the amplification tree can be built on top of that. So after the Z pairs hybridize with the target RNA, the preamplifier binds to the top of the ZZ pair. And on those preamplifiers, you can have multiple amplifiers that further bind multiple label probes. Now, these label probes can either be a chromogenic enzyme or a fluorophore, depending on the assay you choose. And it generates a signal detectable either under a standard bright field microscope or a fluorescent microscope. So this particular signal amplification strategy gives you that high sensitivity and allows you to visualize your target RNA as a single dot, where each of these dots represents an individual RNA molecule, which can be further quantified. So how do we give you the specificity, or how do you achieve specificity with this assay? Background is eliminated because this signal is dependent on both of those Z's binding next to each other on the target sequence. So if both of these Z's do not bind, there won't be a preamplifier that will sit on it, which will, therefore, you cannot form a stable hybridization, and this amplification tree won't be built. And consequently, no amplification of any non-specific hybridization will occur, generating either little to no background signal. So a standard RNAscope probe, if you order, will consist of about 20 of these Z pairs pulled together that are designed to hybridize next to each other along your gene of interest. However, for a tremendous amount of giving you that tremendous amount of amplification and signal potential. However, you only need about 3 Z pairs to bind to the target RNA sequence to give you signal, which is really important for customers that are working with samples such as archival FFPE samples where your RNA can be degraded. So taken together, the combination of our probe design and our signal amplification will really ensure you that high signal to noise ratio, a bottleneck for a lot of traditional ISH techniques. So I talk about this RNAscope technology, but we do have various assays that fall under this technology. The 3 main assays that fall under this technology is first the RNAscope assay. The RNAscope assay is about 20 Z pairs per target, which gives you that single molecule detection and is ideally used for targets such as mRNA or long non-coding RNAs, or any RNA sequences that are over 300 nucleotides. We have singleplex, duplex, and multiplex versions of these assays that can be used either on automated platforms or can be used manually. The next technology that falls under that RNAscope branch is the BaseScope assay. This BaseScope assay consists of about 1 Z pair or can be made up to 3 Z pairs and allows you to look at targets such as splice variants, short or highly homologous sequences, and also point mutations. So targets that are between 50 to about 300 nucleotides. And this is available as either a singleplex or duplex chromogenic assay. And lastly, the newest assay that just came out is the microRNAscope assay. The microRNAscope assay has a new probe design strategy, and this will allow you to detect targets such as ASOs, microRNAs, siRNAs, or smaller sequences between 17 to 50 nucleotides. And this is currently available as singleplex chromogenic assay. So really, with these 3 unique assays under that umbrella of RNAscope technology, you can target RNAs anywhere from 300 nucleotides to 17 nucleotides, kind of covering that broad array of sizes of different targets. So what is our product portfolio? As I mentioned before, we have our RNAscope chromogenic assays in either singleplex or duplex, which can be performed manually or on our automated platforms such as Ventana or Leica BondRx. We have our RNAscope multiplex assays, fluorescent assays, where you can look at up to 3 or 4 targets. And then last year, we came with the RNAscope hiplex assay that allows you to look at up to 12 targets simultaneously on a single tissue section. Our BaseScope assays target shorter sequences, such as splice variants, as I mentioned before, and this is available as singleplex or duplex chromogenic. And then I mentioned our newest assay was the microRNAscope assay, which is available as a singleplex chromogenic, targeting sequences from 17 to 50 nucleotides. But keeping in mind all of these assays, the one thing that is also really important is our probes. Currently, we have over 26,000 probes in catalog. That being said, not everybody may have probes that are in those catalogs. So if you are interested in a probe that's not already available, ACD can design that probe for you, and it can be for any target and any species, as long as you know your sequence of that particular gene. And it can be done in as little as 2 weeks, which is really important and significant in doing studies where you need a rapid production or rapid development of these probes in crucial times. An example would be the COVID-19 probe in outbreak of COVID-19 where you need to do rapid research. So with all that, we do have a lot of applications of RNAscope. You can visit our website, or we have a lot of webinars focusing on different applications. Our top application areas are cancer, neuroscience, stem cells, infectious diseases. And there's a lot of material available on this out there. But the focus of today's webinar is going to be on how researchers are using RNAscope in pain medicine research or in pain research. So I've picked up a few examples of recent publications that came out showing the utility of this technology in pain research. The first one is something we're very proud of. Now, this paper, it came out of University of Texas, where researchers from Theodore Price's lab, they performed a study to understand the underlying mechanism of pain that was associated with viral infections. And they did that by exploring the role of interferon signaling in inducing nociceptors within the dorsal root ganglion, or DRG. So the interferon alpha and interferon beta were shown to induce nociceptive hypersensitivity response in mouse models. So these researchers, they confirmed these findings by performing single-cell RNA sequencing on mouse DRG, and they identified expression patterns of interferon receptors. In this case, they used the RNAscope multiplex fluorescent assay, and they validated their single-cell data, and they visualized the co-localization of these interferon receptor genes with neuronal markers in the DRG. Now, a lot of you may not know this, but this RNAscope image also made the cover on the issue of Journal of Neuroscience. And what this paper is really demonstrating is the role of the RNAscope assay in visualizing cellular activity in cell type specific manner, really giving you power and information to your entire research. Now, this is another paper that came from Karolinska Institute. They recently identified the transcriptional regulator PRDM12 that can possibly cause congenital insensitivity to pain in humans. And they found that the inactivation of this PRDM12 resulted in a complete absence of nociceptive neuron lineage, while proprioceptive and mechanosensitive neurons remained unaffected. So they used RNAscope to confirm the knockout model of PRDM and also visualize neuronal specific expression in these developing DRG. Overall, the results of this paper provided information to the researchers on a potential target for therapeutic approaches aiming at the modulation of these pain perceptions. In this paper, as you see, they actually used the dual-ish IF workflow, where they could simultaneously look at the PRDM12 RNA target along with the TRKA antibody on the same tissue section. I will discuss more about this workflow later in the slides. Now, pain, as you may all know, is a multidimensional experience. And how much the pain is bothering people, it significantly impacts the sufferer's quality of life, especially when people are going through it. The kappa opioid system, functioning through these kappa opioid receptors, or KORs, they are known to contribute to the depressive and the dysphoric states. But whether it contributes to the emotional component of chronic pain is poorly known. So in this paper, researchers from UC Irvine and UCLA, they show that these KORs are sufficient to drive the emotional part of chronic pain that is argued to significantly impact a patient's quality of life. The antagonists to KOR can act as a therapeutic adjuvant to alleviate this emotional component of the pain and improve the quality of that patient's life. So in this case, they used the RNAscope multiplex fluorescent assay to examine the KOR expression in a mouse model of chronic pain and connect the expression of this KOR to its function on a tissue context. They also saw that OPRK1 expression was increased in response to pain and was primarily localized in Th positive dopamine neurons, once again being able to localize expression of a certain target with a certain cell type specific, in a cell type specific manner. This is another paper. So a brush of a feather, or a pinprick that you see. Now, all of these touches or sensations can be perceived as distinct because they're detected by discrete cutaneous sensory neurons. Your body has these specific neurons that detect these sensations. Now, inflammation or nerve injury can disrupt this sensory coding. It can basically mess that system up in your body, and it can result in a maladaptive pain state, including mechanical allodynia. So, when sometimes you like brush your hair and you get pain, so it can disrupt that coating in your body, and it can cause those inflammation can cause that coating to change. And it can result in the development of pain in response to an innocuous touch. So in mice and in humans, loss of mechanically activated piezo2 channels was known to was resulted in the inability to sense discriminative touch. So in this particular paper from Howard Hughes research facility, the scientists showed that the optogenetic activation of piezo2 expressing sensory neurons induced nociception in mice. Now, they used the RNA-scope multiplex fluorescent assay also to compare the ablation efficiency of piezo2 in 2 different knockout models. And they saw that in the second knockout model on the image, the piezo2 HOXP8, that the ablation efficiency was much higher compared to the one in the second when they compared these 2 models with the wild type. Now, the results from this research indicated that piezo2 mediates inflammation and nerve injury induced sensitized mechanical pain. And they suggested that targeting piezo2 might be an effective strategy for treating mechanical allodynia. Really, once again, showing the use of RNA-scope in studying or understanding pain therapeutics and understanding targeted treatments. So these were a few academic examples. I also wanted to show you an example of a pharma application by a biotech company. So ASOs, or antisense oligonucleotides, as you know, are short, about 16 to 20 nucleotides, synthetic single-stranded nucleic acids. They can bind and modulate target RNA. Now, these ASOs can act at the RNA level, and they're very selective. They're highly selective, so therefore they make ideal candidates for targeting traditionally undruggable targets, especially in the case of neurological disorders, including chronic pain. So this is a paper from Ionis Pharmaceuticals, where researchers demonstrated the delivery of these ASOs to both spinal cord and the DRG. And they showed that they exhibit a longer duration of action in the pain system. They used, again, the RNA-scope-ish and IHC protocol. They used a NUN antibody along with MALAT1 RNA target to demonstrate that these ASOs suppress target RNA in afferent fiber neurons in a dose-dependent manner. They saw that the MALAT1 was downregulated specifically in NUN-positive neurons with increasing concentration of treatment, as you can see on the screen, and that MALAT1 ASO exhibited activity in afferent fibers of all sizes in a dose-dependent manner. This is really showing the application of RNA-scope in sort of the biopharma industry. And so I showed you these examples of how customers used RNA-scope with antibody. And that's something we've seen an uptake, a higher uptake by researchers, because they are interested to study both RNA and protein simultaneously on the tissue context. And so that's where I'd like to now divert your attention to this new integrated co-detection workflow. Previously, our customers that did perform dual-ish IHC ran the RNA-scope assay first and then followed it by the antibody detection, and then looked at both the RNA and antibody in the same tissue section. But what we noticed was with that previous workflow, there were a lot of antibodies that were losing their signal due to the pre-treatment that you have to use for RNA-scope assay. So ACD developed this new integrated co-detection workflow that allows for a more extensive screening of compatible antibodies, and you don't lose, or it allows you to retain that protein signal after doing your RNA detection. So it works, the workflow starts with permeabilizing your cells, then binding your IHC antibody to the protein, hybridizing your ISH probes for RNA detection, then doing the amplification, as I talked about, building the amplification tree, then adding your secondary antibody, and finally visualizing both your RNA and protein on the same target. And this is a really, really powerful technique with a lot of applications in the field. You can correlate your RNA and protein expression. You can also examine cell type specific gene expression. A lot of our customers and some tech companies out there have been using RNAscope to validate their newer antibodies on that tissue context. You can visualize cellular localization of RNA targets. You can also discriminate antibody detection of multiple targets, such as like splice variants or highly homologous sequences. And in lieu of the big era of infectious disease research that's going on, we've seen researchers use this technique to detect pathogen at different replication sites with the host cell markers. So with that, this really creates a powerful workflow for customers to get that bigger picture, combining both RNA and protein on the same tissue and giving you a lot more information out of your experiments. So with this, I'd like to now switch gears and talk about a new technique that came out last year and is being widely utilized in the field of not only neuroscience, but a lot of other transcriptomic studies. And that's the RNAscope high-plex assay. As you all may know, in the last couple of years, single cell research has really rushed up. A lot of people are doing transcriptomic studies to reveal that complex and heterogeneous nature of these complex organs, such as the brain and the tumor heterogeneity and a lot. And they're identifying a lot of novel cell types that are characterized by more than one gene. So it's really presented a need for a technique that allows you to look at multiplex data, but do it on a spatial context. Because a lot of these discovery transcriptomic tools use dissociated cells. And so you end up losing that spatial and morphological context to that information. So what is the RNAscope high-plex assay? This assay allows you to provide signal amplification of up to 12 mRNA targets on a same tissue section. This is done by 3 iterative rounds, where each round will look at up to 4 targets at a time. And at the end of each round, you use our image registration software to register these images together and look at these 12-plex data individually or combined at a single cell level. Now, between these each round, you do go through a fluorophore cleaving procedure, but our validation data shows that there has been minimal to no effect on your RNA and tissue morphology in this process. Currently, this assay is compatible with fresh frozen and fixed frozen sample types. But we do have a pipeline where we will enable this assay for customers to use on FFPE sample types as well. And what it's really important for is for customers that have precious samples where they don't want to run multiple rounds or 4-plex assays. So they can get a lot of data from a single tissue section of this assay. So to show the application of this particular technology, we utilize this paper that came out of Stanford University by Gopsha and his fellow researchers that was published in Cell Reports in 2016, where they identified novel cell types of D1 and D2 striatal medium spiny neurons, or MSNs, by analyzing 1,200 striatal cells using single cell RNA sequencing. So this is the workflow application, right? Researchers use these discovery tools such as PCR, single cell RNA sequencing, where they're using dissociated cells, and they're getting a lot of this data, right? So in this case, these researchers identified discrete cell types that existed in a continuous spectrum of transcriptional states and found that this functional diversity within a complex tissue arised from a small number of discrete cell types. But they didn't actually confirm these results, nor did they show the spatial relationship of these D1 and D2 subtypes. So we employed the RNA-scope assay to confirm and spatially map these subtypes in the mouse brain. For the purpose of this application, we utilized 3 different experimental panels. We used the HiPlex 8 reagent kit that allows to look at 8 questions simultaneously. And then we also, 8 targets simultaneously. And then we also looked at the RNA-scope HiPlex 12 assay that allows to look at 12 targets simultaneously to kind of resolve this medium spiny neuronal heterogeneity. This is an image from the RNA-scope 12 assay. As you can see, we were able to visualize all of the D1 and the D2 targets or the cell types on a single tissue section, right? But you look at all of this data and you see there's like a lot of information going on. So question you may ask is, how do I read this? How do I know what target is reacting with what in all of this mumbo jumbo? And that's really where our image registration software comes into play. So to show how the utility of the image registration software works, the researchers at Stanford, they highlighted an important finding in which they identified a group of cells that express both the D1 and the D2 genes. So using our HiPlex image registration software, we identified this D1 and these D2 genes by toggling on and off my interest of genes using our image registration software. And this is really, really like a great tool that allows you to individually select for your genes of interest and then resolve the heterogeneity of these complex tissue types. This slide is like a graphic that was created using online tools, showing you the power of this assay and our image registration software, and allowing you to visualize the heterogeneity of these cell populations on, again, that single tissue section. But this is just showing you an application of this technology, but I did want to show you an example of how one of our early researchers that used this assay applied it in pain research. So now the molecular information of projecting neurons these neurons, they carry this pain and itch related information, but how they did that was unclear. So scientists from UCSF, they performed RNA sequencing and they revealed this transcriptional diversity of these spinal and trigeminal parabrachial projecting neurons. So they performed the HiPlex assay that I just talked about, along with FOS labeling to reveal dysfunctional heterogeneity of these neurons. And they also in this paper actually compared the mRNA expression to protein. And they suggested that some of the trends they saw may reveal a true difference between the rate of gene transcription along with the translation of that particular target. Really, again, showing you the power of this new technique. As you can see, they were able to utilize less samples, but get a lot of data out of that publication or the paper. So with all of these applications in pain, I wanted to kind of divert our focus more into the demands of the current field. As you all know, we are going through this COVID-19 pandemic, and so a lot of labs have focused their attention to COVID-19 research. So I did want to get you some time of yours to show you the applications of RNA-scope in COVID-19 research. We have a variety of products and assays available out there that can enable you to study the pathogenesis for COVID-19 viral detection across a broad range. You can utilize our singleplex assays to look at the virus alone, or you can utilize our duplex assays to study the virus to host interaction, the virus receptor interaction. You can also utilize our multiplex assays to not only study the virus receptor interaction, but as you all know that post-COVID, there is this cytokine storm that's been observed in patients. So you can look at that immune response by multiplexing your experiment. And lastly, you can also look at some histopathological markers, really highlighting the need for an assay that allows you to look at your data on a tissue context and look at the changes that happens in patients pre and post-infection. And so with our specificity, sensitivity, and the morphological context, you really can study this broad range of viral pathogenesis. I also wanted to introduce to you the ACD's new probe sets. On this slide, you're seeing probe sets for COVID-19. These were targets that were chosen based on early literature that enables customers to buy these sets and start their research. We do also have probe sets for applications in neuroscience, and you can look at those and get more information on those on our website. The probes in these bundles are in pre-assigned channels, so it prevents you from going through that one extra step of assigning probes in different channels, and just kind of gets you jump started on your research projects. So now I wanted to show you a few examples of how RNAscope was used in COVID-19 literature, some early papers and some new papers. And really, in a broad category, the applications of RNAscope in COVID-19 were primarily in these 4 areas. It was to study cell and tissue tropism. It was to understand natural immunity, to sort of enable vaccine development. And this is a really exciting one that I will share later. And lastly, to study placental transmission. 4 areas that are not only important in COVID-19 research, but really in any infectious disease research. We've seen utility of RNAscope even in the past during the Zika virus outbreak, or even in HIV research. Really applications along all of areas of infectious diseases, but with focus today on COVID-19. So let's start with the first one is why were researchers using RNAscope in natural immunity, right? To study natural immunity. The questions that were being asked was, can natural immunity be induced by infection? And you may have all seen that in news. There's a lot of discussions going on, is if a patient infected by COVID-19, are they building this natural immunity to reinfection? So a lot of the questions being asked in the publications were those. And RNAscope was being utilized to visualize the tissues and the cells that were infected with SARS-CoV-2, but to also look or demonstrate any viral replication sites. So one paper that I chose to show you today is this paper, and an understanding of the protective immunity to SARS-CoV-2 is extremely critical for both the vaccine as well as public health strategies that are aiming to end this global pandemic. So like I mentioned before, the key unanswered question was whether SARS-CoV-2 infection resulted in a protective immunity against re-exposure. So this paper is from Harvard Medical School, where the scientists developed a rhesus macaque model for SARS-CoV-2 to study the pathological and immunological findings of COVID-19. They also used this model to study natural immunity against SARS-CoV-2 after re-exposure. They used the RNAscope chromogenic red assay to visualize the SARS-CoV-2 viral RNA in lung tissue, along with detecting its replicating intermediate. And that's really a big power of this SARS-CoV-2 probe by ACD, is you can not only look at the viral strand, but you can also look at the replicating strand and find out the active replicating stride on the same tissue section by either performing a duplex assay or a multiplex assay. In addition, they also performed IHC staining with cytokeratin and the viral nucleocapsid protein from Novus, which is a sister company of ACD, to indicate the infection in alveolar epithelial cells. So in summary, a lot of this information will further help scientists to test for vaccines against the virus and also enable for development of therapeutics, really allowing them or giving them power to further their research. So what is the next area that I talked about was vaccine evaluations. And this is something that, that everybody's kind of excited in the field. They're all ready to find out when it's coming out, and has a really strong urge in enabling this research. As you may all know, there's over 160 vaccines in development, But the big question is, how well do they work? So in these two papers, the RNA-scope assay was used by the researchers to visualize the viral infection in control animals, but also confirm the absence of virus in these tissues after the animals were treated with the vaccine. And so for this particular, I took an example of the vaccine that a lot of you may be aware of, is the Moderna vaccine. Now, the effect of this severe, or the SARS-CoV-2 on viral replication in both upper and the lower airways was important to evaluate in non-human primates. So in this paper, the researchers evaluated the efficacy of this mRNA 1273, which is the Moderna vaccine, encoding for the spike protein of SARS-CoV-2 in the non-human primate species. And they used the RNA-scope chromogenic assay to detect the viral load in lung tissues after vaccination. So as you can see, compared to the control group, they saw a significant decrease in inflammation on administration of 10 microgram and 100 microgram of this vaccine, really kind of enabling them to confirm their findings and confirm the efficiency of this vaccine on a tissue section. And the last one that we saw was the use of RNA-scope in studying placental transmission. So this is a study where Luisa Patani and other researchers, they studied 2 cases that were SARS-CoV-2 PCR positive mothers, neonates and placenta. And they used the RNA-scope LS, which is a Leica Brown assay, to visualize the virus in the placental tissue and evaluate the tissue distribution while still retaining that morphological context. So the RNA-scope assay validated the presence of this SARS-CoV-2 RNA in these infected placentas, supporting the possibility of a vertical transmission of SARS-CoV-2 from mother to baby in utero, a study that really helps researchers identify or understand the prognosis of mothers, pregnant mothers that have been tested positive with COVID. Another example of this that I wanted to show is this particular image that came out of Arcana Labs and Yale School of Medicine, where also researchers presented a case of a COVID-19 positive mother in her 2nd trimester that was complicated by severe preeclampsia and placental abruption. So they really wanted to distinguish and differentiate between the physiological effects of preeclampsia and COVID-19, again, for better prognosis of pregnancy. And they used the RNA-scope manual Brown assay to visualize the SARS-CoV-2 RNA in the placental tissue. In addition, they also went to compare the staining pattern of the SARS-CoV-2 with the ACE2 receptor, which is being now known in literature to be the receptor that SARS-CoV-2 interacts with. And they found that this pattern closely matched the ACE2 receptor pattern as seen on the Human Protein Atlas. So we've really seen a broad application of RNA-scope along different needs or answering different questions in the research field. And I've only shown you the tip of the iceberg, as there are a lot more publications supporting these applications in the field for infectious disease research. But going back to our general applications, the growth and adoption of our technique this RNA-scope technology is really best exemplified by the number of peer-reviewed publications. Our first publication came out in 2011, and since then we've had over 2,700 publications that have been published in numerous journals. And we see a lot of these being in like high-impact journals such as Nature, Cell, and Science, really showing the power of the data that these researchers are getting. Now, it is, as I talked before, RNA-scope is highly relevant across multiple fields of research. Our top areas are cancer, neuroscience, and infectious, but we've also seen applications in stem cells, HPV, development, inflammation, immunotherapy, and a lot of other applications that can't even be bucketed. So if you want to learn more and kind of understand more of some of these other applications, you can go to our website or contact our support team, and we can give you more targeted publications to show you the utility of this assay. So I hope today with all of this data on the technology, the publications, COVID-19, and on some upcoming and new technologies, I've convinced you today that Autoscope uses a unique probe design strategy to dramatically improve your signal to noise ratio. We currently have over 22,000 probes on catalog, but like I said before, if you know your sequence of the gene of your interest, ACD can design any probes for any target and any species within 2 weeks. Autoscope can provide you both quantitative as well as spatial information on your target in the complex tissue environment at a single cell resolution. Our assays such as BaseScope and microRNA-scope, you can detect splice variants and short sequences down to either 50 nucleotides or 17 nucleotides respectively. And lastly, Autoscope assays can also be complemented with IHC using our new integrated co-detection workflow to allow you to simultaneously look at RNA and protein on the same slide. So with that, if you have any more questions or if you need any more information on this technology, you can contact our support team or visit our website. Thank you so much, everyone, for your time and for attending today's webinar. Thanks again and stay safe.